Hello and welcome to the University of Edinburgh's Research Insights. Thank you for joining us. My name is Hazel Lambert, I'll be your host, and I'm joined this afternoon by Dr. Megan Davey, Dr. Alewo Idoko-Akoh, and Jen, Nicola and Nick, who are off camera but making everything work in the background. Megan and Akoh, welcome.
Hello.
Hi.
Hi. So Megan and Akoh are going to tell us all about their research this evening, but first of all, a little bit of housekeeping. This event is being recorded, but all of your cameras and mics are off, so we can't see you, we can't hear you, so don't worry if your dog barks or the doorbell rings, it won't be on our recording. Remember, this is your chance to ask our scientists your questions. So the Q&A box is open. You can ask them anything you like about what you hear this evening. Now, we are very lucky to have an incredible space called the Easterbush Science Outreach Centre at the university. It's fully kitted out in exactly the same way as all the other research labs, but it's not for our scientists, it's for you. It's usually full of school pupils and other visitors to campus, and our plan had been to join you from there this evening, but given the new lockdown restrictions, we decided we're going to play it really safe and stay at home. So almost all our research is still going ahead in the labs. So Megan and Akoh do usually still work there during the day. That gave us a chance in December to do a little bit of early sneaky recording just in case this happened. So we're going to use Megan's video that we made in December in place of the live demo. And that means that you'll still see what we had hoped to show you from the lab. We're going to be talking about chickens and chicken embryos this evening and how they're used in genetic research. So you will see a developing embryo inside an egg. And this kind of animal research is really tightly regulated in the UK. And remember, you can ask us absolutely anything you like about what you see and hear in response to the presentations this evening. So Megan, if you get yourself ready, Megan is a research group leader at the Roslin Institute and the Royal Dick School of Veterinary Studies. And in her work, she builds maps of the connections between the genes involved in limb development. And then she uses what she learns to create new research tools. And she's going to tell us about it now. Megan, over to you. Thank you.
Hi, everybody. Thanks, Hazel. I'm really pleased that you could join us today. And I'm sorry we're not joining you from EBSOC, but now my kitchen is quite comfortable as well. So today I'm going to just really quickly talk about the things that I'm interested in as a scientist. Okay, so my lab is really interested in understanding the really sort of fundamental rules of embryonic development. And that's not just for chickens, that's for us and for all other vertebrates as well. So I could be interested in all sorts of different parts of the developing embryo, the brain or the pancreas or the bladder or anything. But the area of the developing embryo I've chosen to focus on is really the developing limb bud. And that's because people have actually been studying it for quite a while. So I'm not starting from scratch. We actually know quite a lot about how limbs grow, but we definitely don't know everything. Now, the reason I've chosen the developing limb is because it's just a really simple pattern, and trying to understand how that pattern forms is really important, not just to understand how limbs grow, but just to really understand all the kind of very fundamental ideas about how an embryo grows. So by concentrating on understanding this pattern, so my pattern is just that we've got how do we make 5 fingers, or in a chicken 4 toes, and how you make a thumb at one side and a little finger at the other, we understand lots of the rules of how normal embryos develop. And sometimes we discover new genes which are important in embryonic development. And also because the genes that we use to build an embryo also get used throughout our lives, these genes are actually important for human health right from conception all the way through to death, probably. Okay, so my big question is really, how do we make 5 fingers, and how do we make a thumb at one side and a little finger at the other? And that's just the very simple pattern. what we do know is that there's a gene called sonic hedgehog, which is extremely important for making the correct number of fingers and toes. So how do we know that? So sonic hedgehog, so this here is a picture of a developing chicken embryo, and I've done an experiment on it so that we can see where the sonic hedgehog gene is turned on at this particular point in time. Now, although every cell in an embryo has all the copies of all the genes, they don't use them all the time. They only turn on genes when they really need them. And here you can see sonic hedgehog. So all the cells which are blue have turned on sonic hedgehog at this point. So this is the developing chicken, this is its head, this is its eye, this is the developing face. And this blue line that runs down here is the developing spinal cord. And you can also see that there's this little white outgrowth here, that's the developing wing. And in the posterior side, those are the side that's going to make the little finger, these cells express sonic hedgehog. So how sonic hedgehog comes on and off and is turned on at exactly the right level is really important for making the exact number of fingers. So what I do is that I take different chicken breeds or avian breeds as well, like emus or kiwis sometimes, and we can look in their genome because you don't even need to have embryos to look at a DNA sequence these days, it's all online, but we can also look at their morphology. And what I'm really interested in is when these chickens or animals have fewer fingers like the emu that they should have, or sometimes more. So all of these, so a normal chicken foot should have 4 toes. This is a silky chicken foot, like if anybody keeps chickens or they've seen them, they're the really fluffy chickens, have a look at their feet next time because they actually grow an extra toe. So silky chickens have a very small change in DNA, just one base pair. That means it's not actually in the sonic gene itself, it's actually in the instructions to turn on sonic hedgehog only in the developing limb bud. And these guys turn on sonic hedgehog, they just turn it on too strongly. Likewise, emus, we know, don't turn on sonic hedgehog strongly enough. This guy up here, this is a special chicken breed which is only kept at the Roslin Institute, actually has nothing wrong with sonic hedgehog itself, but actually can't receive the sonic hedgehog correctly. So understanding how sonic hedgehog turns on and off in the limb at the right time, in the right place, at the right concentration, and we want to try and ideally have this kind of sweet spot of sonic hedgehog to make the correct number of digits. Understanding that is really important for understanding how sonic hedgehog turns on and off all the way through life. So I'm going to show you an experiment very briefly, and this is kind of typical of the work that I do in the lab, and especially at Roslin, where we have lots of special breeds of chickens, some of which we've made ourselves, including transgenic lines of chickens which express fluorescent proteins. So this is an experiment to show how sonic hedgehog really works. So sonic hedgehog is a protein, and cells use it. So these cells which turn on sonic hedgehog use it as a signal to talk to other cells. So the cells in this side of the limb bud, the sonic hedgehog expressing cells, are using sonic hedgehog to tell all the other cells in the limb bud how many fingers they should make. And also that they should make a little finger on this side and a thumb on that side. So the experiment to really show that is that I can take the sonic hedgehog expressing cells out of one chicken, and in this experiment I've taken them from a red chicken, and these cells, as well as expressing sonic hedgehog, also express the tomato protein, so they're red throughout life. I can see where they go, and I've transplanted them into a live developing embryo which is expressing the green fluorescent protein. So here you can see the little ball of sonic hedgehog expressing cells I've put in there, and this is on the wrong side of the limb. This side would not normally see any sonic hedgehog protein, and if you let this embryo grow out for a few days, you get a limb that looks like this. So a normal foot should have 4 toes, and this one has 3 extra toes that I've marked with an asterisk here. Now the interesting thing is it's not the sonic hedgehog or the red cells which themselves have gone on to make the extra digits. You can see that because the red cells are here, and that's just on this side of the limb bud. They've actually told the green cells that they should make more digits. So that's just an example to show how important sonic hedgehog is, and that cells, groups of cells, use it to tell other groups of cells too. Okay, so this is an example of the experiments that I do in the lab, and now Nick is going to play a movie, which is me in the lab in December, showing a group of students how I open eggs and how I do the experiment. So over to you, Nick.
To study embryonic development, it's useful to be able to study embryos. Sometimes we also use cell culture, and that gives us a lot of information, but you can't really regenerate pattern in a bunch of cells in a dish, or not very easily. So the place where pattern and development really happens is, of course, in the embryo. So that is the best place to study it. We could study all sorts of animals, but chickens lay eggs, and they lay them all the time. So they give us a really constant supply of fertile eggs, but they also, it means that the embryo is prepackaged in a little self-contained container, the egg outside of the mother. So if we wanted to study mammalian development, that just is hugely complicated and ethically different as well, because the embryo is inside another being. But chickens, they're outside, and not just chickens. I also use different birds. I use eggs from different species sometimes. So I think that's a rhea, for example, but mostly I use chickens. And this is what I do. So when I go into the lab in the morning, this is more or less what I do every morning. So I take my fertilized chicken egg, and I'm going to say now, what I'm going to show you is not, you won't normally find embryos in supermarket eggs that you eat. So don't worry. Supermarket eggs come from farms where they don't normally keep the boy chickens with the girl chickens, if they keep any boy chickens at all, where these guys are kept in pens with males and females. So we get lots of nice fertilized eggs. So this egg has been incubated for 3.5 days. So the embryo here is just beginning to make a little limb bud. It's got a beating heart. It's beginning to undergo some neurogenesis. It's decided which part's going to be the forebrain and different parts of the brain. But anyway, this is how I open an egg. So if you've ever looked at an egg, you'll see on one side it has a blunted end, and on the other side it's a bit more pointy. And if you've ever eaten a boiled egg, you'd know that there's like a little gap, a little air sac that's not filled with egg white, and that's always at the blunt end. So I kind of utilize this to really get into the egg without damaging the embryo on the way in. And it's really not very complicated. So I'll probably do it on the other microscope. So just using a bog standard pair of nail scissors, you can just very carefully make a little hole. I think I might be okay this time, Jane. And actually, this sometimes goes wrong, as you can imagine. That's why we're lab coats. You know, you can hold the egg too tight and it can break, or it could be just a little bit fragile. I've only ever had 1 or 2 rotten eggs in my whole career, which is quite lucky, I think. Okay, so now that gives me access into the very top of that air sac at the end. But the embryo is still lying underneath a few more membranes which protect it. So what I'm going to do is I have a little bit of saline here. I'm just going to drop a little bit of saline on the top. That just gives me a chance to put my forceps in without really damaging anything. I'm going to go in there and make a little hole, and the saline will suck through to the embryo, and then I can take away the white membrane without touching the embryo at all, because we don't want to do that. It would be really easy to accidentally break a blood vessel at this point. Okay, so there we go. So now I have an embryo in its egg when the limb bud is starting to develop, and I could go in and I could do lots of different experiments on this guy. So but it's still quite small. It's actually much bigger than a mammalian embryo. That's another advantage of chickens. But the other kind of special thing is, so people have used chicken embryos to do embryonic development studies for like 100 years. And you become very good and specialized at this, and you use microscopes. But actually what we have at the Roslin Institute, which is really special, it's one of the only places in the world that has these, is that we have transgenic embryos. So these are embryos, or actually chicken lines, where we've inserted transgenes. So I'm going to show you 2 different lines today. One has got GFP, so green fluorescent protein, which has been randomly inserted into the genome. But actually this GFP chicken line expresses GFP in every cell. And we also have some TD tomato chicken. So TD tomato is a red fluorescent protein, and it's also a transgene. But I'll show you the tomato ones first. So I'm just going to swap over to our microscope camera, hopefully. Share screen. There we go. Okay, let me just get this guy in focus for you. Okay, there we go. So here, if you look carefully, you should be able to see, well, I wish I had a pointer actually, maybe no, I don't. So here we have, oh, there's my forceps. Oh, sorry, chicken. So there you can see this is its head end, and you can see maybe there's a little grey circle there. That's its eye. And if you look very carefully, it's just done a heartbeat there. The heartbeat is just there squeezing. Sorry, it went out of focus when I put the forceps in. There we go. And you can hardly see it because it's really quite small, but this little blob of white stuff just there over top of the beating heart is actually the developing limb bud. So the body is like, this is the head end, and then this is mostly the neck, and then this is going to be the trunk, and its leg is down here. So it's pretty good. We can do lots of experiments on this, but it's even better when we turn the lights on. So I'm going to literally turn the lights on now. So this is an excitation that will excite the GFP. Oop. I might just turn that down a bit. It's so bright. Okay, so that is super bright now. Every one of those cells is expressing GFP, and yeah, we can use these embryos to do all sorts of experiments, like mixing cells between the red and the green embryos. So that's the GFP guy. I'm going to actually put the TD tomato one on because literally the GFP one is so bright it's quite hard for you guys to see it. This is another one. So this is an embryo of the same stage, but now instead of green, oh, come on, embryo, sit in the right place. This one is red. And actually this fluorescent protein is super bright as well, but the excitation I have is not quite perfect for TD tomato, so we're only seeing about 5 to 10% of the actual total protein. I'm just going to turn it up for us. Oh, he's also green. Why is that? I think it's actually just a strange shadow. Okay, but here maybe we can see the limb bud a little bit more clearly. Focus in just a little bit. So now you can really see the heart beating just at the top of the screen there. I hope you can see that. Oh, where's my forceps? There's the heart beating. And then this little white blob here, that is the developing limb bud. And down here, that's the leg bud. So it doesn't look like much at this stage, actually, the limb bud. It's just a blob. It's sort of hard to know if it's got any pattern or anything. But in actual fact, at this stage, pretty much all the cells in that limb bud know what they're going to make in the final limb. They already know which bits are going to be the humerus, the radius ulnar, and which are going to be the digits. It's just that none of those cells have actually really fulfilled their destiny just yet. They're still kind of early on, but they already know what they will be. And one of the reasons that we know that is because from experiments that we do, swapping the green and the red cells between embryos, is that we can take like a tiny little piece of red where we think the digits know that those cells will become digits, and you can put it into a green embryo and you can watch, and those red cells will always make digits. They will never go in and make anything else. So that's one of the ways, or one of the experiments that we do with these chicken embryos.
Brilliant. I have to tell everybody, we filmed that in our EBSOC, in our Easterbush Science Outreach Lab. We don't normally have cuddly cells in the lab, but we put it in there just to brighten it up for a bit of decoration for everybody. Megan, thank you so much. Do you want to say anything to finish off before I introduce Akoh?
I was just going to finish off by sharing one more slide. So I have to say, when we took that video, we were using like an old iPhone and it was pretty good, but I'm just going to share my screen and show you just a slightly clearer picture of what an embryo looks like in an egg. I'll just share that quickly. So that's not a chicken egg, that's an emu egg. There we go. Oh. So that gives you an idea that these are my fingers here, and that's the developing chicken inside. On that note, thanks very much, everyone. I'll be here afterwards for questions, and I hope that we get to see you sometime next year at one of our public events.
Brilliant.
So
Thank you so
note,
much.
I'll go back to you.
Akoh, are you there and ready? If you switch your camera on and your mic on.
Oh, hi, hello.
Hi, great, brilliant. Thank you, Echo. So while you get yourself organized, ready to share your screen, I'm going to introduce you. So Megan and Echo work at the same place, the Roslin Institute. Megan's told us all about how she watches chicken toes form to learn more about what controls the way limbs develop overall. Now, Echo is going to tell us more about his work on ways to breed chickens so that they don't catch bird flu. And this is really important because it can help stop new versions of flu from spreading into people. Now, Echo is a vet originally, and then he went on to do a PhD in a research technology called genome editing. So he's one of our double doctors. So Echo, when you're ready, over to you to share your screen. Thanks.
Yeah, so hello once again, and many thanks for the opportunity to speak to you. So in this presentation, I will show you how we use genome editing at the Roslin Institute to study bird flu in chickens with the aim of developing new antiviral strategies to keep chickens happy, healthy, and free from the threat of bird flu in general. So avian influenza is also known as bird flu, and this is a disease of birds caused by the influenza A virus. So this particular virus, let me see, this virus circulates in wild birds, and wild birds can actually spread this virus to other wild and domestic animals, such as the pig and chicken. And recently in the United Kingdom, in Britain particularly, there has been a legal requirement for all captive and farmed birds to be kept indoors to prevent them from making contact with wild birds. So humans really get bird flu virus from wild birds. However, humans can get bird flu directly from chickens and other types of poultry. In chickens, bird flu causes a very deadly disease. And in humans, the most severe pandemic in recent history is the 1918 flu pandemic, which is believed to have come from birds. So you should note that bird flu in humans is different from the usual seasonal influenza for which you get the flu jab for every year. There are no human vaccines for bird flu. So humans have a very, very poor immunity to bird flu. And so when bird flu gets into humans, it can be very, very deadly. So if we can prevent bird flu in chickens, then of course we can have healthier chickens, and it will also be a very good strategy for preventing human infections. So scientists believe that some chickens may carry genes that prevent them from catching bird flu. And this is because when there's an outbreak of bird flu in a chicken farm, a few chickens often survive these deadly outbreaks. And these survival chickens may carry genes that prevent them from becoming sick with bird flu. So there's a great interest in finding out what genes or variations of a particular gene help prevent chickens from becoming infected with the bird flu virus. So in 2016, scientists at Imperial College London identified a chicken gene that is important for the bird flu virus to successfully infect a chicken. So we at the Roslin Institute collaborated with these scientists and discovered the variant of that chicken gene which stops bird flu virus from multiplying. And so we made this discovery by performing experiments in chicken cells that we grew in the lab. But we had one big problem. We didn't know if this particular gene variant was actually effective in preventing a real chicken from catching bird flu. So to resolve this issue, we have to make a real chicken that carries that gene variant. And so to make that real chicken, we use genome editing technology, and we have developed this technology at the Roslin Institute. And so once we've made this genome edited chicken, then we can go on to test this chicken to see if they'll get bird flu or not. So to make genome edited chickens, we use these special cells called primordial germ cells. So these cells in adult chickens will go on to form sperm and eggs. And we obtain these cells by collecting a very, very small quantity of blood from young chick embryos like this one you can see in the egg here, which we incubate for up to 3 days. So we perform this procedure using a special glass needle. And so I'm going to play a video for you right now to show you how we get the blood from the chick embryo. So you can see the heart of the embryo beating in that video. And so these chick embryos are really, really young, and they do not feel any pain. And these procedures are actually regulated. So we only perform it only if it is absolutely necessary. So we collect a very small amount of blood, a microliter of blood, which could contain up to 20 of those cells. So next, we have to put the embryonic blood we have collected into cell culture plates that contain a special growth medium for these cells. So when we perform these cell cultures, we perform them in sterile safety cabinet environments like these ones. And so this is done to prevent contamination. And so we put the cell cultures, of course, which is in these plates, back into an incubator set at 37 degrees Celsius. So this is important to keep the cells healthy and growing. So we have to keep monitoring the health of these cells by looking at them under a microscope. And so that's what I'm actually doing in this particular photograph here. And what you can see on the right is a photograph of these cells that have been taken under a microscope. And so we have to keep observing these cells to ensure that they're healthy and dividing. We then have to make sure that we have the required number of cells before we go on to perform genome editing. So we perform genome editing using molecular scissors such as CRISPR-Cas9. So these molecular scissors actually cut DNA at a specific location that we desire, and we're able to carefully design them to go to any specific location that we intend. And so in this image here, I'm holding a reagent of CRISPR-Cas9, and which, as you can see in that tube, is colorless. So it is invisible to our eyes. So most of these procedures are completely invisible. We can't actually see what is going on. So in the photograph below, I am performing a process called transfection, which will put the CRISPR-Cas9 molecular scissors into the cells so as to achieve genome editing. And so we then need to know whether or not the genome editing has worked in the cells. So we extract DNA from these cells, which we have targeted using CRISPR-Cas9. And then we send the DNA to another facility where a process called DNA sequencing will be performed using a machine such as this one. And the result that we get back looks like this. So you can see in this particular result, DNA is made up of these 4-letter codes, A, C, T, and G. And in this result, an A has been changed to a T. So this process is not 100% effective. So we have to keep trying multiple times until we get the desired results. And so we then have to use those genome edited cells, which I've illustrated here, to make a chicken. And if you remember, I told you that these cells go on to form sperm and eggs in the adult chicken. So we obtain fertile eggs from special types of chickens that we keep at Roslin Institute. And so we incubate these eggs for 3 days. And then we carefully create a small window in these eggs. And then we then put the genome edited cells into these embryos. So these embryos are actually surrogate embryos that are sterile and do not contain their own germ cells. And they are going to be of the same age as the same embryos that we got the germ cells from initially. And so once these eggs are put back into an incubator, they're going to hatch. And then once they hatch, the chicks grow into adults, which we then have to breed together. So these surrogate chickens contain sperm and eggs that have been formed from the genome edited cells that we initially made and injected into them. And so once we breed them together, then we obtain genome edited chicks, which we can then use for our studies. And so finally, once we have our genome edited chicks, of course, they grow into adult chickens that we can even breed to produce more chicks. But what is more important is that we can now perform our clinical trial on these genome edited chickens by directly infecting them with a specific dose of bird flu virus to see if they are protected or not. And so if that gene variant that we have discovered, and then introduced into these chickens through genome editing protects the chickens from bird flu, then this gene variant can be bred into other chickens around the world to improve their overall welfare and health, and also protect humans from bird flu. So this is an example of how we use genome editing as a research tool to study bird flu and also other chicken diseases, with the overall goal of developing new ways to keep chickens happy, healthy, and also protect people. So thank you all for listening.
Thank you very much, Akoh. Megan, do you want to put your camera and your mic back on? And we'll move over to questions from our audience. We've got 2 already. But Megan, you know exactly what I'm going to ask you for the first thing. You have to tell everybody why that gene is called Sonic Hedgehog.
Okay, so the Sonic Hedgehog gene is not just in us, it's in mice and fish and all sorts of other vertebrates, but also invertebrates as well. So the gene was actually first discovered using fruit flies. Now, the fruit fly geneticists used to take groups of fruit flies and just sort through and look for interesting looking phenotypes, interesting funny flies that didn't develop normally. And then they would collect them and they would figure out what the gene was. And so they found the hedgehog gene, it was called, because in the little fruit fly maggot, it misses out every second segment. And the segments that it keeps are like, they have little bristles on them. So basically they look like little hedgehogs. So that's why the gene was called hedgehog. We actually have 3 different hedgehogs. And of course, it was inevitable that it was going to be called Sonic Hedgehog. Actually, 2 labs found it at the same time. One lab called it vertebrate hedgehog, so super boring name, it didn't stick. And the other group called it Sonic Hedgehog, and their name stuck.
Oh, and I understand there's now a committee, isn't there, that decides what genes can be called? Because, yeah.
There is a very official committee, Hugo, which names genes, especially for humans. And I have to admit that lots of clinicians don't really like the name Sonic Hedgehog, because Sonic Hedgehog does go wrong in lots of different congenital disorders. And then, you know, people don't like saying, oh, I have this problem because of Sonic Hedgehog. That sounds stupid. So.
Yeah, fair enough. Okay, well, we have questions coming in from our audience. And remember, if you're watching, you can upvote questions. So if there's something that you really want to ask, you can put a thumbs up on it and then I'll make sure it gets answered. So let's start off with a question about, so Megan, this one's for you. Are there any ethical issues associated with using fertilized eggs?
Yes, there is. So any experiments that are done on animals in the UK are really tightly regulated. Embryos are not considered animals until they reach a specific stage of development. And that's a bit different for all embryos from different species. But for chickens, we're not allowed to do experiments and then hatch the chickens. They have to be, the whole experiment has to finish 2/3 of the way through embryonic development. So that's day 14 for a chicken, because it normally takes 21 days to hatch. And if you wanted to do an experiment like Akoh's experiment, probably where you wanted to see what happened to your transgenic chicken, then you have to apply for ethical approval to the Home Office.
Yeah, so that's the Home Office of the United Kingdom. They have the overall responsibility. Thank you, Megan. Okay, question for Akoh, slightly different topic, but I can see why lots of people would be interested in this. And this is from Finley. And he says, how did you go from being a vet into this kind of research?
Oh yeah, that's a very good question actually. So the thing is, I still consider myself a vet, but it's just I somehow got tired of just being in the clinical setting, you know, every single time and then seeing the same type of cases over and over again. I just wanted to be part of any kind of, you know, global effort that is trying to solve, you know, the kind of diseases and challenges that we see in animals. So it's a difficult transition, but I think I'm happy about that. It's been very good so far.
And that's a really important point, actually. You know, this is a global challenge. This is something that affects people wherever they live in the world. And even, you know, right now, anyone who keeps a chicken as a pet has had to bring them indoors or create a sort of anti-bird net. You know, tell us why it's so important. What happens if bird flu runs rampant in a chicken farm?
So bird flu is really, really deadly. In many cases, when you have an outbreak of bird flu, I mean, all the birds, all the chickens in that surrounding area have to be killed. And I mean, so you have to think of it from 2 different aspects. So first and foremost, it causes, there's this huge economic loss for the farmer, and of course for the government, because of, you know, they lose taxes and profits and things like that. But then the second issue also is that bird flu can get into humans and cause a very deadly disease. I mean, in fact, up to 50% of times, I mean, when humans get infected with bird flu, they actually die from it. And the most serious pandemic in recent history, like I mentioned before, is actually the 1918 flu pandemic. I mean, much deadlier than the current COVID-19 pandemic, and it's believed to have come from birds. So it's actually a disease to be taken very seriously. And since we know the virus circulates in wild birds, and so to prevent that jump from wild birds into chickens. And since a lot of chickens mingle with people, it's important that we actually take this, prevent that contact between wild birds and chickens, which are very, very close to humans.
And on that note, do you know if a chicken could catch COVID-19?
Oh, that's a very good question. So I'm not a coronavirus researcher, but I've read at least 2 papers that have experimented to see if that was possible. But we know that the particular receptor that the coronavirus uses, is kind of different in chickens and in humans. So to date, we have not seen any evidence that the coronavirus can actually infect chickens because of the difference of the receptors between chickens and humans.
Okay, thank you.
Yeah.
I'm going to ask you one more career question that's come in from Heather, and she wants to know if it's possible to be a part-time vet and a part-time researcher. Can you do both?
Oh yes, there are a number of vets at the Roslin Institute that are actually still in clinical practice. So they do some kind of locum shifts in the weekend and then still come into Roslin to do research. I mean, yeah, it's really, really possible. I mean, at Roslin, you have so many examples of that going.
Brilliant, thank you. And it's the same for our medical doctors as well. Lots of them do research and work in the clinic too. All right, Megan, we've got loads of questions that are coming in about Sonic Hedgehog and about genes. I'm going to go with another question from Heather. So she's asking, does the Sonic Hedgehog gene have a role in triggering apoptosis? So that's cell death when a cell dies during the development of limbs.
Yes, it does. Actually, good question, Heather. So the limb does everything it can do to make sure that it has the right amount of Sonic Hedgehog, not too much, not too little. So the first control is that genetic control, switching it on at the right time, switching off at the right time. But even then, if there's too much Sonic Hedgehog in the limb, somehow the cells know that and they actually kill themselves off. So the Sonic Hedgehog cells are really sensitive to actually how much Sonic Hedgehog they themselves are making.
Okay. And a follow-up question from that, are birds the only practical species to perform gene editing?
Oh, no, not at all. So Akoh might be the best one to answer that one, I think.
Yeah, that's a very good question. So we're able to perform gene editing in birds because we can grow their germ cells, the one I talked about in the presentation. So we have not been successful at growing germ cells properly in other bird species. But of course, in the future, it will be possible to perform gene editing in other bird species, but it's definitely possible to do gene editing in other bird species, using other methods.
Okay.
Yeah, we also do gene editing in pigs and in sheep as well.
And there's
Yes.
a farm at Roslin, isn't there? There's where those things.
And other research institutes do them in other animals as well. I think even Drosophila and C. elegans, all sorts of different.
So Drosophila is a fly and C. elegans is a worm. You'd be amazed how many worms and flies are used in research. Right, next question is from Lauren Wood, and she says, how would the rollout of a genome-edited chicken occur, and what does it mean for breeders? So she's thinking about how it would be applied to birds bred to a standard of perfection or for non-industrial settings like small hatcheries. So if you got to the point where you had a genome-edited chicken that you knew was resistant to bird flu, how would it get into the population, as it were?
That's a very good question. Something that we always talk about. So the first thing you have to understand is that usually we have this issue of patents. So if you have a genome edited chicken, usually there's an intellectual property issue associated with the genome edited chicken. Usually most researchers will want to have a claim on that particular, discovery. So which means one of the best ways to roll it out will be through commercial partners that have a breeding platform, a breeding strategy. So, they have these so-called breeders, and then they're able to multiply them successfully and then push them around the world. That's a question that I think chicken breeders, the top chicken breeders can answer. For local chicken keepers, I'm not exactly sure how that's going to happen. I guess that's going to be a government effort. Yeah.
Can I'm going to add to that as well. So I think another interesting thing is that we don't necessarily have to always use genome edited chickens. we as scientists use genome editing in the lab to see whether we could make a resistant chicken, but then knowing which is the right gene and which are the right changes in the DNA that would be desirable to have in the chickens, we could also then look in all the chicken breeds around the world to see if there's any naturally occurring chickens which have those changes just by chance, just by nature.
And this is where it all fits together. Genes control development. You're looking at development. The fluorescence that we saw, that's showing you which genes are present in the embryo. That's right, isn't it? So it all fits together in stages. And we've got another question here about it says, could this process be used to this process of genome editing to tackle viruses in chickens? Could it potentially be used on humans, or would this be seen as inhumane?
That's a very
it's a very relevant question, and I think it's seen as inhumane. I can't see that happening, but it has actually happened in China a few years ago. Children were genome edited in order to be resistant to, well, supposedly to make them resistant to HIV, and generally there was a massive outcry around the world, and that's not the type of science that I think any countries want to pursue.
Yeah, fair enough. Okay, there's some other questions here about polydactyly. I hope I pronounced that correctly, Megan.
Yeah.
How would you use the developing limb experiment to provide answers for the reasons for polydactyly? And can you just say what it is as well for people watching who won't have heard of it before?
Yeah, so polydactyly, poly is just many and dactyly is fingers. So polydactyly is extra fingers than your normal 5. And it is one of the most common birth differences that we see in humans, in fact, that we see in all animals. So the work that we have done and many others really have shown that most extra fingers are probably down to a little bit too much Sonic Hedgehog or a little bit too long during embryogenesis. We understand many of the genetic changes that might have caused those. And what my lab is interested in doing now, so there's, I think limb differences, congenital differences, are just, we're all a big, variable of different phenotypes. I mean, it's just one fantastic part of the spectrum, I think, having a little bit of an extra finger, a whole extra finger, several extra fingers. There's no way that we want to prevent that necessarily, but I think what's useful is trying to understand perhaps what happens in the limb in your hand when you have those extra fingers. So do you grow extra muscles as well? Do you get extra nerves? Do you get extra tendons? So that's what we're working on at the moment, actually, is trying to understand if you have an extra bone that grows, what happens to all the other soft tissues, and do they grow normally? Do they not grow normally? Because having normal nerves and muscles is actually that's important to know how those grow for kids with extra fingers.
Okay, we have time for 2 last questions, so I'm going to take 2 from the Q&A box. The first is for you, Megan, and that's, how have you managed to carry on your research during lockdown?
Actually, lockdown was really good for me because my group had done a lot of lab work, but I had been a bit slow in writing up papers. So there's always really useful things that we can do sitting at our computers. Writing up our research papers is one of them, but also it's really useful as well. I mean, there's so many resources online. You can look for new genes in the genomes that are published. There's thousands and thousands of genomes online. That's all the sequences of all the DNA from thousands of different species. So that's one of the things that I've done during lockdown, and also just talking to colleagues on Zoom as well.
But our active lab research is going on as well, isn't it?
Yeah, that's true. That has gone on, but we've been quite careful to make sure that if we can work from home, we do. So I have been at home more than the people in my lab who work in the lab more. And we wear masks and we wear other PPE and we stay apart from each other, and we have a very organized rota about who goes in and when they go in.
And then, Akoh, I'm going to ask you the last question, and this is actually a bit cruel because it's a really big question and there's only about a minute left. But what I'm going to do, everyone, is I'll send an email out when the session ends, and I'm going to put a link in it to how CRISPR-Cas9 actually cuts the DNA. So, Akoh, if you want to give us a very brief explanation of how CRISPR-Cas9 actually cuts the DNA in the lab, if you want to have a go at that.
Yeah, so think of CRISPR-Cas9 as a complex that has an instruction and a scissors. So we designed the instruction to find the specific part of the gene that, or the DNA that we want to cut. And then the scissors is made up of protein that goes to that location to cut that DNA. That's just what CRISPR-Cas9 is. So an instruction and the scissors. Go, find, and then cut.
Akoh, you need a prize for that. That was amazing. Really well done. Thank you both so much. Thank you everyone for watching and for all of your questions. One of the things that we really want to do with this series is be able to cover the kind of research that you watching would like us to. So I'm going to put a slide up in a couple of seconds that has a code to a website called menti.com. If you follow that code, you'll be able to just type in your subject suggestions, what you'd like us to cover. I will be back on the 27th of January with research scientist Dr. Elaine Emerson and surgeon Mr. Ian Nixon, who'll be talking about their research on throat cancer. So thank you everyone for joining us. Thank you, Megan and Akoh, you were fantastic. Thanks to the rest of the communications team for running the webinar in the background. Take care, stay cozy at home, and I hope you can all join us in the future. And I'm just going to put that slide up now.
Bye, everybody. Hope to see you sometime this year in person, I hope.